Okay, so we've talked a little bit about this before, but let's kind of get into the nitty-gritty of how sperm is produced, and we'll talk about how eggs are produced. So the testes are divided into these small subdivisions, and they're called semifertile tubules, where sperm is produced. And each tubule is lined with the ability to make these things called germ cells, which is just a fancy way of saying stem cells that make half the genetic material of the male. Serosine cells promote sperm production, and that is going to be this area where we can make these cells sort of go into half, and we can start to go ahead and make them into normal sperm. It has a blood testes barrier, and it's sort of a tight junction between these Sertoli cells, and they're a really tight junction because we don't want these cells to go off into the normal circulation because they're half and they wouldn't work anywhere else. They'd have to be fertilized to continue on. If we look at sperm production on itself, you could see that how it works is we have a normal cell. So this is going to be, it's called 2N because it's got the normal complement of genetic material, which is 2 copies of every chromosome. And it sort of stays on the outside of that blood testes barrier, so they can produce these things called daughter cells, which are going to eventually be these cells, these sperm cells. What will happen is they'll pass through that layer, and they'll start to make our secondary cells, which are going to be starting to divide in half. We have our primary spermatocytes. Eventually, we're going to split this in half. That's why I went from 2N to N. That's going to be our secondary spermatocytes. And eventually, we're going to prep these cells each to become sperm, and that's called spermatogenesis. And that's going to be where the cytoplasm of the cell will start to grow, and we'll start to get the tail and everything that we sort of think about. So really big picture here, let's not get too crazy with this one, is you go from your normal complement of genetic material, you start to divide that in half, and then you make those into clones. That's why we went from this cell to this cell, and we start to make all those into sperm, and then eventually we do spermatogenesis, where we're going to create that little area for the front of the sperm and the tail, and we'll name those in a second. If we were to look at a piece of sperm or a cell, you'll notice that we have this huge flagella that basically works like a boat propeller. We have a big area of mitochondria. The mitochondria are just areas that receive that fructose and are designed to make tons of ATP, and that ATP is designed to make this flagella spin, kind of like a boat. We have a nucleus, and at the end of our nucleus, we have this little sort of area, and that's going to be called our acrosome. And our acrosome is a chemical that's going to contain an enzyme that can penetrate the egg. And once one sperm has hit that egg, the egg changes on the outside. So even though a lot of viable sperm could be hitting that egg, as soon as that first one hits it, it's kind of closed for business for the rest. So if we have another sperm that comes in 2 seconds later, it's too late. If we have another sperm that comes in 2 hours later, the outside of the egg has changed so much that it can't penetrate through, even though it has this chemical. In eggs, we'll do eggs in our next video.